Excited that everyone's able to join us today. This is a panel discussion from the Global Virus Network on the H5N1 avian influenza issue. I'm Dr. Maggie Bartlett. I'm a virologist at Johns Hopkins Bloomberg School of Public Health and program director for the GVN. I'm really quite honored to get to moderate this esteemed panel of experts and discuss the complexities of H5N1, surveillance, and the global impact. Our panel includes experts across virology, genomics, epidemiology, and disease surveillance and pandemic preparedness. And they each focus on a different aspect of understanding and addressing viral implications for global and human health security. We're going to review some recent data on transmission and pathogenesis, assess some different control measures, and discuss strategies for global pandemic preparedness and response, and provide actionable recommendations. Active participation from both the panelists and the audience is highly encouraged. So we want to collaborate to address this viral threat and enhance global health security. If you would like to speak on a topic that we are discussing, please connect to Ruby Ferrari in the chat, and she will add you so that you can talk or put your comments directly in the chat. I'm now pleased to introduce our panel of experts. Dr. Peter Palese will be joining us later. We have Dr. Elodie Gideon, who is renowned for her expertise in viral genomics and bioinformatics at the National Institutes of Health. We have Dr. Martha Nelson, hello, who is a specialist in pathogen evolution at the human-animal interface, also at the National Institutes of Health. We have Dr. Jared Evans, who's known for his expertise in molecular virology and emerging infectious disease modeling and preparedness at the University of Nebraska Medical Center. And Ms. Lauren Zauer, who is the director of the Special Pathogens Research Network and has expertise in public health emergencies and outbreak response, also at the University of Nebraska Medical Center. And Dr. Albert Osterhaus, who is a renowned virologist and known for his expertise in many zoonotic viruses and contributions to global infectious disease research and pandemic preparedness, affiliated with the University of Veterinary Medicine in Hanover. Yes, and there's Dr. Peter Palese, who is internationally recognized for his pioneering work in influenza virology and vaccine development at the Icahn School of Mount Sinai. And so each of our panelists brings really a wealth of experience to today's conversation, as many of the registrants, and we look forward to the insights across the board and recommendations as we look at both the challenges and the opportunities surrounding H5N1. So I've prepared several questions to help us guide the discussion. And I would like to start off with how advancements in genomics and bioinformatics specifically can enhance our understanding of H5N1 transmission dynamics. And I'll probably start this off with Dr. Gideon with her expertise, and then allow each different panelist to comment if they would like, and then anyone in the audience.
Hi. Thank you, Maggie. And I will punt this at some point to you, Martha, because you've been doing the molecular epidemiology of this outbreak. But as probably most people know on this panel, and of course, you know, the audience, genomics has really helped advance our understanding of various outbreaks. And we've seen it, of course, with COVID, but flu is also followed very closely with genomics. And what we were able to see very quickly in doing a lot of the sequencing is also these reassortments that happen. And I think, Martha, you have shown in some of your trees potential reassortments, or we know the origin of the history of the different segments of the virus. And so nowadays, doing routine sequencing is really a non-issue, and so we should be able to track this outbreak very closely if samples are available to do so. So I think there's no limitation in capacity, no limitation in analyses, and what's very important and seems to often be lacking when there are outbreak investigations is the metadata. Exactly, the date of collection, the site of collection, And that seems to be the most important in the tracking of these outbreaks. And so, Martha, can you speak to what you've seen?
Yeah. I mean, just to follow up exactly with what Elodie was saying, we saw during SARS-CoV-2 how much could be done with genomic data to trace at very fine scales transmission between different kinds of demographic groups, between countries, even between rural and urban areas. And if you have very intense genomic sequencing, we have the tools, the Bayesian phylogenetic tools. I mean, we have this really rich methodology for extracting information from the genomic data. As Elodie said, it's really important to have the metadata that goes with that genomic information. With cattle, we're using genomic data in a lot of different ways. We're doing it to look at spatial transmission across states, when this outbreak likely emerged using a molecular clock, how long it's been transmitting in cattle undetected, for it seems many months based on the amount of genetic diversity we're seeing, how it's spreading between states that are pretty far from each other, Idaho and Michigan and North Carolina, and how that epidemic is linked across all these different states. But then also looking at adaptation and how this has changed since it's jumped from birds, how it's adapting, to mammals, and what it takes for this virus to be fit in a new host where we've really have never seen influenza, other than at least influenza A, transmit with such high fitness. But then also even just really basic questions of prevalence. I mean, we have reported cases, but we're starting to use wastewater and genomics to kind of figure out where this virus is and what, how much, again, genomics kind of allows us to see a black box. There's a lot that we're not seeing in this epidemic, and it gives us kind of clues into seeing, what the scale of this is.
Thanks for that, Martha. And Elodie, do any of the other panelists have comments on this and the genomic prevalence that we're seeing?
Well, let me just say one thing. We have H5 viruses for quite some time, at least 2 decades, where we have been confronted with H5 viruses. And there were a lot of, the only thing which stayed sort of the same is really the hemagglutinin. And there has been a lot of change in the non-hemagglutinin genes. And with the H5N1 in cows, we are really, we would love to know what is really different, that this comes in others and in organs which we really haven't predicted or haven't really been confronted. So I think it will be very important to also find out what these genetic changes are and whether we can correlate that in any way with the transmission of this virus, which is really a big, big mystery, I think.
No, I think that's a really excellent point to highlight, that it's been prevalent and around for the last 2 decades, and to the extent that it's circulated in these different populations, we're really just starting to understand, it seems. Albert, I see your hand is up if you want to comment.
So I wanted to make the same point as Peter was making, obviously. So this virus has been around since 97, of the last century, but these are completely different viruses. What is important is what Peter also stressed, is to really look at the genetic makeup there. So apart from reassortments and the like, but look for the specific mutations that are going to make the virus possibly transmissible. And we'll come to talk later about pathogenicity. One of the things that intrigues me tremendously is if you look at the pathogenicity of this virus, well, basically for cattle, it's not very pathogenic. But if you look in cats, for instance, yeah, and we have been doing some experiments lately in ferrets as well. It's not at all the same as the original virus that we found in Hong Kong or the one that came from Indonesia. Pathogenicity has changed dramatically, but I think the most important thing is to look at transmissibility at the moment. And there are markers for that, although we don't have them all, obviously. But I think that's an important point.
And the point to follow up on your comment is also the fact that there are specific mutations that have been identified in some of the polymerases that are not what we were expecting, but could be mutations that are providing this adaptation in the mammalian host. Right, Martha, I remember you showing.
I was just going to jump in and say, I mean, we've been, as you say, we've been studying H5N1 for decades, and we've been cataloging the mutations that we expect to see in the HA and in the polymerases, you know, these E627K and PB2 and, you know, the sialic acid receptors. And we have a whole list of things we expect to see, and none of those mutations are in this cattle virus. So it's really kind of, you know, we have some candidates, you know, this, you know, position 631 and, you know, some others, but this is, you know, this is really changing what we know about influenza and what it takes to transmit in mammals. And that, you know, door has just opened and those experiments have just begun, but they'll be really important.
But
it also shows the challenges of predicting phenotype from genomics. I don't think anyone would have looked at this virus and said, this is going to do great in mammals.
No. But also when you look at what's happening with the pathogenicity of the virus, you know, so it has increased tremendously for some of the carnivorous species. And basically we initially thought about this one or two papers on one from Martin Beer about a similar virus in cattle, but giving barely anything. But in principle, why there are so many unknowns there? Because when we look at the whole series of mutations that you see, we haven't got a clue what is really making the difference there in terms of both pathogenicity, but could also influence transmissibility. You know, so I think those are the key questions. And the other thing is we are talking about genomics, but one of the things that has not been done in the US, there's a lot ongoing at the moment. But if you look into Europe, for instance, there's no seroepidemiology. Just the classical things have not been done yet. And the question is what is ongoing in the US at the moment in terms of seroepidemiology? Yeah, because it's fine to characterize all these viruses, but you would also like to know what is ongoing there. If you look at the different states, is the virus more broadly spread than we know today? And I'm not aware of things happening there. We are starting up to discuss these things in Europe at the moment, but we are far behind, of course. So basically, it would be good to see what is going to happen in the US in terms of seroepidemiology.
You said you're behind. You're ahead. You don't have it. I think it's wonderful.
You know more than me, Peter.
There's no, I mean, is there any H5 in cattle in Europe? I thought it's not.
We haven't seen it yet, but I think
it
is important to be vigilant there. If this can happen in the US, we have birds all over the place. You know, Holland, Germany, there's migratory birds all over the place, and there's cattle grazing outside all over the place. So it's really a warning that this could be much more far-reaching. And in US now, it's really understood there is a serious risk there. And WHO, ECDC, CDC, we're all alerting the people, but we don't know what's happening in Europe, where we have a very intensive farming as well. And you're completely right, it hasn't been found, but nobody has looked into it.
Yeah, that was my question, is it that it hasn't been found or we haven't looked? But
well, I know in some countries they're starting up to do some seroepidemiology, but there's no concerted action, so to say. We don't have a CS, we don't have organizations that you have in the US, whereas our farming is just as intensive.
May I ask a question, Dr. Osterhaus? Maggie, can I ask one question,
yes,
please?
Absolutely.
So what is your, if I put you on the spot, how do you think the H5 is transmitted in cows?
That's the $1,000 question, yeah. So basically, there's two options which might coexist. I do not exclude the possibility that it is being transmitted through the other as well. Yeah, so really the suction cups and that it's really contaminated and spreading like that. Although respiratory spreading cannot be excluded.
So those two, the machinery and respiratory.
that for me, if you look at the extent of what's ongoing, those are the two most likely mechanisms.
Mm.
And then, of course, and that means that also when you're transporting animals from one place to the other, you bring the virus in as well. But that is we don't know the full pathogenesis yet, although some papers have appeared now. But what's your opinion, Peter? Because you put me on the spot.
No, you're an animal. You're much more an expert in animals. I'm just a mouse doctor, that's all.
I'm gonna pivot and put Lauren and Jared on the spot so they get a chance to answer this question, and particularly with their expertise, I think it'll add another dynamic to what we're discussing.
Great.
Well, I wanted to jump in there, but that was a good conversation. So I wanted to pull the thread on what Dr. Polizzi said about looking at the non-structural proteins, because understanding the dynamics of the polymerase can link then to what Martha was saying about function, genomic sequence to function. And we don't have a really fast, quick polymerase assay that can be commercially available. We don't have one that can identify error rates or mutation rates really quickly. So that's like an assay development thing, which might be a little farther down in the discussion line. But that's something that in order to really link those types of things would be really useful. There are other assays that could be built too, but primarily we look at the HA, and we have really good assays for that. But being able to link the sequence to what they're actually doing, we don't have those assays yet. And so to be able to, we always say, the mutation rate of influenza is 10 to the minus 4, but we don't know that if that's for every single flu that's out there. And it's hard to test that. So it's not something we can do in mass, but if we had an assay that could do that, you'd be able to link those types of error rates and be able to build dynamic models that Martha's really awesome at. So that's another thing that could really be looked at, would be useful in these types of events.
But there's a lot of full-length sequencing going on, isn't there?
Yeah.
Okay, there should be some clues, as we have learned from the COVID pandemic, but also from the swine flu. Yeah. So basically, I agree. If you had rapid tests to do that, it would be great. But the capacity you have in the US to do large numbers of samples and to do full-length sequencing there, which when you find clues, you can focus that obviously, and then. But I fully agree that that's besides the serology, I think that's the thing that we should focus on.
Yeah, I might just add to Albert's point about the surveillance. I think, we're looking where we're seeing indicators that it's already there, right? And so expanding surveillance into nearby animal reservoirs that we know to be true, but maybe haven't looked, expanding surveillance into more systematic approaches to the human populations that might be immediately affected. I think CDCs and USDA very recently have started taking the steps to do that, but that requires coordination, collaboration with industry. And so, putting the effort to making sure that happens is really important. And I would also add that the, our neighbors to the north and south of us in Canada and in South and Central America have potential high-risk exposures, right? And so looking in those areas and finding partnerships through North and South America are essential right now.
Thank you. I think that's a really intriguing aspect to add, that we need to be looking widely and making sure that we're bringing everybody to the table like industry partners.
Could they ask, is there any, I don't know about that, is there any trafficking of cattle or goats, sheep back and forth between Europe and the US? I'm not aware of that, but is there used to be, but I'm not sure if that's still happening, because that would be another risk to contemplate.
Yeah, we've talked about this, and one thing is that they're not lactating cattle. You're not flying lactating cattle across the ocean. And so we still don't know whether lactating cattle are the only transmission risk, but right now they're believed to be the highest.
Yeah, okay.
So they certainly are getting moved into Mexico and Canada and other, neighboring countries, but they're probably not being flown overseas.
Yeah. And the other important thing is to consider semen as well. Probably the virus will not be in the semen, but we don't know it's in the milk as well. So and semen is definitely being trafficked back and forth.
Yeah, I also wanted to loop back to the comment about South America and what Elodie had also mentioned about reassortment and in terms of the risk for Europe, because these viruses that are getting into cattle, these are reassortants between the Eurasian and America's lineage. So right now they're only found in the Americas, in North America and also in South America. Right now they're not in Eurasian birds or wildlife. Whether, that doesn't mean that other variants of this virus couldn't jump to cattle. But this particular lineage is not found in the Eastern Hemisphere.
That sort of addresses, if you can see in the comments, Adolfo put in a really interesting comment, and I wonder if that's the reassortant, the fact that it's a reassortant is why it found its evolutionary pathway into cows, which is what he says in the comment. How did it make it into cows? Flu has been seen in horses before, didn't make the jump into humans, but here maybe it will remain a cow virus, but maybe it won't, right? So if we can figure out how it made its way into having the cow as a good host, could help us understand whether it'll do the next jump.
I think that's a fantastic point and a really great comment. And I think it's, again, one of the things that adds to why having these discussions is so integral. And so in the interest of time, I'm going to pivot to another question on if there are key challenges in developing effective vaccines against specifically what we're seeing here. Do we see it as a risk? What innovative approaches should be explored or are being explored? And I might let Peter tackle this first.
Let me defer. Who wants to go first?
Well, Peter, you should do that. You're the champion there. Yeah, we could fill in later.
Give me the question again, please.
Sure. Just what are the key challenges to developing an effective vaccine, or do we feel that the vaccines we have currently would be effective against what's circulating? And is it getting where it needs to be? So are the people who are at highest risk getting vaccinated, or are there other innovative approaches that are being explored to try to contain this?
So you're just talking human vaccines now, is that correct?
No, so human vaccines or vaccines in livestock.
Because that's two completely different things, of course. There's other
Yes.
Maggie, you're not asking one question, you're asking 5 questions.
I will defer to which question strikes you as the most important at this time.
I mean, I think in terms of making a vaccine against H5, I think it's easier than making a vaccine against SARS coronavirus 2, because we have made vaccines against H5, and I don't think we have anything like long COVID, or we don't have any real complication as we have, and we know much more about H5, about influenza viruses in general, and I think also in terms of H5 viruses. So I don't think this virus is immune suppressive, and so I would feel that in general, I think we should be, first we have very good drugs. I don't know how many kilograms we need to protect the cow with Tamiflu. I think that's another question. But we do have good antivirals, which we don't have against SARS coronavirus 2. And we also have, I think, a virus which we know better than SARS coronavirus 2. So that I would like to say that this is a somewhat easier problem, than what we had to do with SARS coronavirus 2. And yes, I think you asked the question, are there any breakthroughs? Sure. I mean, I think the platform of mRNA is certainly something which works in flu, and I think is an advantage in terms of speed and all of that. But my take-home message is I think it's a somewhat easier problem than the more recent SARS coronavirus. So.
No, I think that's a poignant point, and I see several hands up. I saw Albert's first, so I'll let him tackle.
Well, I think I agree with Peter. We know flu vaccines for a long time. We've been making these vaccines for almost 100 years. So basically, if you go for the short term, the classical vaccines, like preparing for an H5N1 pandemic, just before the swine flu came, we and others have done clinical trials already up to phase 2. Phase 3, with candidate vaccines produced in the classical way, and you can have that ready in 3 months. You can even do it in Baculo and whatever platforms you have there. So that would be an easy one. Then we can of course go for all the other platforms and see if those have benefit. But at least for the short term, we can in the next 3 months we could make a vaccine. We already did so for the H5. For instance, vaccinating people in Taiwan and other places, so in the hotspots. So that's one side of it. I think, and with the new technologies, obviously there are some advantages, are some disadvantages. So that should be looked into very carefully. Vaccinating cattle is another thing, because the price is another thing. It can and does it really give you protection against also transmission? That's the other thing. A word of caution here, Peter mentioned the use of antivirals, kilograms of antivirals in cattle. I think I would be strongly against using antivirals in animals, because that would increase the potential to generate escape mutants there. So we have discussed that also for H5 when the tigers got infected and we had big problems in Thailand, and we decided not to do it because it would create another opportunity for the virus to escape. But I think for the vaccines, I fully agree with Peter that we know the technology and we could explore new technologies as well. But it's important to first define what do you need the vaccine for, and the priority for me would indeed also be the vaccines for humans there. Over.
Maggie, I'll jump in afterwards. Just to take us a little ways away from the technologies. I think we do know, I agree with both Peter and Albert, we do know the technologies very well. What we've lost is the public's trust in vaccines. And so I think, as we scale up vaccination strategies, it's important to work with, our behavioral scientists, our social anthropologists, and our community members to make sure that people are willing to participate in the last phases of the trials that need to happen as we move from bulk into vaccine ready to go, and that we continue to be transparent about the new technologies, work with our media, work with our academic partners to share information about how the vaccines are being developed. I think this will be especially important if we have any challenges with the egg supply needed to make traditional flu vaccines. And so, I think everyone's excited about what mRNA technology can do, but we do have to be really cognizant of the fact that there is a global lack of trust in vaccination right now that we're really working hard to combat and to build that trust back up. And so the technology and the advancements in the science can really only be one part of the focus. And I would just argue that we need to make those resources available to sort of community health workers and public health practitioners right now to ensure that we don't make a ton of vaccine that is not used.
Those are really important points. Go ahead, Jared.
I'm gonna combine the two discussions that just happened there a little bit. And we do have a national stockpile of H5N1 vaccine. One of the things that can easily be done, this has been introduced into people. We can take those people and see what their serology is, or inoculate new people and see if it is able to block this currently circulating virus. It hasn't really been done. People have done it with other H5s that have emerged. People have done it with other H7N2s and H9N2s, things like that. But people haven't done it for this current outbreak as far as I know. So that might be able to have that bridge of the technology, what we currently have available, what we would need to ramp up, what new things we could develop, and then the public health side that Lauren.
Jared, how many doses do we have in the stockpile?
Nobody really knows. They don't divulge that information. but it is an adjuvanted antigen. So it's not a it's not a full virus that's inactivated. It's antigen focused. So it is a little bit quicker to make. And it could come out of, as baculovirus or yeast or whatever
And Jared,
they want to use there. So yeah, go ahead.
Yeah, just jump in on the doses. So we have heard the number 500,000 of the old 2007 virus tossed around, or 2007 vaccine tossed around, and we know they have a ton of bulk. I think the question that we've been asking of CDC and asked for quite a bit is how much of that new candidate virus is in vaccines, is in syringes, and what we've heard is a couple 100,000 doses, but it's completely unclear what is making up what's in those syringes. So I think that's a place we could definitely advocate for is understanding what is actually in the stockpile, what makes up those numbers.
Perhaps if I may say something about it. So we were gearing up for, just before the Mexican or the swine flu, we're doing exactly the same thing. And then as a matter of fact, at the end of and there were stockpiles as well, both for antivirals and also for vaccines. And then it became an H1N1 that came from basically from Mexico. And it took a couple of months before we realized what was going on until it came into the US. So I think we have to be careful that we are not focusing too much on those doses that are available, but it's more important to be able to gear up and make new doses, and preferably of using the new strain then. And if it's hemagglutinin, then using the technologies that we have, either egg produced or baculo produced or what have you.
Albert, could I ask you a question about that? Sorry to jump in, Maggie. We've heard that there's been ferret neutralization studies on these new vaccines, and that the data look good, but we haven't really seen the data in humans, and they should have neutralization studies done by now, right? On that, at least on the individual human samples that they have. Do you have any sense of what's behind the holdup?
I have no clue, and I think it would be good if also you can do human studies, but you could easily do ferret studies as well. Yeah, just if you have a series of vaccines, just look at the breadth of protection. We did similar things with the H5 from 2000, from before the pandemic, and as a matter of fact, we saw that if you add the AS03 or the MF59, that you did get very broad protection anyway. Definitely after 2 shots, but even after 1 shot already, the adjuvant is probably more important there than the vaccine itself. So I would at least make a stockpile of adjuvant, because if the numbers are going to be the important thing, yeah. So you get enormous dose sparing and also much more broadening of your response. Yeah. And a lot of work, that kind of work, has been done on MF59 and AS03. I think for flu it may be out because of the narcolepsia, but I think really look into the adjuvant as well. And there's quite a bit of work ongoing there in the US.
No, that makes a lot of sense. I think there's a couple questions in the chat. I see, Lauren, you offered to answer one of them live. So given the reports of PCR positivity of H5N1 of milk products in the US, should PCR testing of milk be prioritized in Europe as well to detect?
I think it would be incredibly helpful to start to do more PCR of milk, especially because even though we're not necessarily looking at live virus, if the milk's been pasteurized, it gives us an indicator of potential presence or past presence, right? And so it might give us, as we navigate the sort of challenges around access to the livestock, it at a minimum could tell us a little bit about what's been there recently and target resources a bit.
And the same can be said for wastewater surveillance, right? Which seems to be a standard way of doing things more and more. We're bringing this more and more. And could we also, Maggie, because I'm looking at the questions, and I had a similar question as one of the people who posted, because we're talking about vaccination and potential surveillance in milk. We haven't yet talked about that human case and the fact that it's not exactly the same, or it's, it's a little distant from the outbreak lineage. And I was wondering if anybody had more information on that. Martha, you have more information on, do we know where that case comes from?
So all we know is from that New England Journal of Medicine case report that came out last week. And it does appear to be, someone who was a dairy worker, someone who had contact with cattle. I mean, it's, seems glaringly obvious that this was a cattle to human transmission event in that northern area of Texas where we've seen all the cattle cases. Why that human virus is such an outlier on the tree is another $1,000 question. And to me, it just
could it be, I'm sorry to interrupt you, but could it be, what do we know about the actual sequence data? Is it high quality? Do we know, is it just?
So they have not submitted those reads to SRA.
Okay, that's what I thought. Okay.
So we don't have the raw sequence data. The actual genome itself looks okay. It's not full of ends. It doesn't look like there's anything particularly wrong. But for us, we've interpreted that as just an indication that we're missing a great deal of data from cattle, especially from early in the outbreak, and that there might have been more, that there are kind of black boxes of data that we still haven't seen, and there might be quite a bit of diversity that we haven't seen yet, including one of the cattle lineages that led up to that human case. We have some good data from multiple states, but I think we're missing large chunks of this outbreak.
Oh, Albert, you're talking, but you're muted.
Thank you. Well, it might be, if I may come back to the epidemiology question. So I think we are talking about PCR, but I think in many countries there are all kinds of programs running for TB and other things, by serological screening for brucellosis and I don't know what. And it might be good to see where we can tap into those. I'm not exactly sure what's still being done in the US. But if you already have the samples, it's a very practical way, to very routinely screen quite a number of animals and farms. So and then serology is much easier, of course, than and much cheaper than PCRs. And as soon as you get a positive hit, then you come in with your PCRs. I think just to see how you manage the crisis, and if everything is negative, then it's fine. Then you don't need to go to PCR testing, I think. So I think this is just a matter of how much money is going to be spent and what is the possibility to pick up positive farms there. And we have a lot of experience in many countries with doing serological screening of cattle, of course.
I think it's an absolutely great idea to do serological screening of cattle. I think the problem is that the economics don't line up in terms of farmers' incentives to do screening of cattle. Especially if they're asymptomatic, if they're, if there's no imminent problem, there's not going to be any incentive to find a high path H5 reportable disease. It's then going to bring people in testing and potentially create problems. So I think that's what's a huge elephant in the room problem here.
But that would be similar for PCR testing, although you only go for symptomatic ones then.
Yeah, exactly. So at least they're symptomatic, at least there's a problem, at least there's, something that they're, you run into the same issues there. People, often would rather just dump the milk and not test. But that's, we're missing huge opportunities for testing because of those misalignments of incentives.
No, I agree. But in certain countries, it will be easier than in others, I think.
Yeah, not easy in the US.
Yeah, so that, thank you for bringing that question up, Elodie. I see that that's from Ann Wiley, and there's some additional questions about long-term infection in the eye and where that case happened, which we already partially addressed. But I think it circles back to what we started talking about at the top of the hour, which is having metadata, having all the information as quickly as it comes out so that everybody can be working to answer those questions quickly. And someone else asked about surveillance in Central and South America, and that in theory it sounds great, but how realistic is it? And is there funding for this in low and middle income countries specifically?
Again,
and I think
funding is always an issue, but a bigger issue is incentives. I think a lot of countries right now don't want to find it. And what's the incentive for them to find it right now from producers? And I know lots of veterinarians, lots of scientists who would like to do the testing, can't get access to samples.
Well, it's always the same with the veterinary field, of course. If you remember the BSE question, yeah. So what happened there? And before we could start testing, well, this was very difficult to get incentives, definitely from the UK, and eventually it would have been much better if we would have been much faster there.
Mm-hmm.
Yeah. So I think if we really looking into the human risk, yeah. So for TB and for brucellosis, etc., programs have been implemented. So as soon as people become aware of a possible problem for humans, I think then it should be mandatory in some way or the others. But I understand quite well that it's a difficult problem.
Yeah, and I think here there is also a misalignment between what's important on the human side and what's important on the veterinary side. I mean, with TB, that's, highly pathogenic in the cattle. So the farmers are highly motivated to screen for that and control it. Whereas here, in some ways, it's the zoonotic issue is more important, at least perceived right now.
So I suppose that's an interesting aspect. I wonder if people have thoughts on.
In Europe, the issue will be as soon as there's a human risk, they start closing borders between countries, and then it becomes a huge incentive. Yeah. So you see that with avian flu, for instance, yeah. Although there's a pandemic ongoing, countries are protecting themselves in inverted commas by closing borders. Yeah.
I wonder if people have thoughts on the comparison between how we handled SARS-CoV-2 and how we're handling this, and what different mechanisms we think should be championed right now at this point in time, as we're starting to see a signal.
Well, COVID was a pandemic, so we are not there at all. Yeah, so basically we are just in the prelude to spread among humans. So it's a completely different situation, obviously.
Yes,
I do think we have a runway that we're not taking advantage of, maybe. If we're talking about potential for human cases, I mean, anytime we see influenza doing things in mammals that we don't expect, we should be paying attention, right? And so the concerns that I've seen and have recently in how things are going, at least in the United States, is really around the regulatory frameworks that allow us to make response activities and including science in that space more accessible. So sample sharing, management of samples, I think, dealing with the select agent regulations, dealing with the category A regulations associated with these samples in the US has so far been a bit of a barrier. And I think we're only just recently starting to see the regulations be adapted so that people can do the work they need to do to better understand what this virus is doing and what is happening, particularly in cattle. I think the surveillance mechanisms need to be spun up. And I take Martha's point completely, that there are disincentives for reporting in cattle, but we have the opportunity to make incentives. And we need to figure out what those incentives would look like for industry. I think just 2 days ago, the USDA made some movement in this space, or 3 days ago, around incentivizing testing. And so, given that the dairy cows in particular seem to get better and can go back into production, what does an incentive look like to understand whether or not they're sick, and getting access to the farms? What does that look like? And how can we ensure that testing alone doesn't change people's viewpoints or uptake in the food chain? And I think that's what, a lot of industry is worried about. They're worried about their bottom line, and they're worried about what testing will do for their production. And so making rapid strides in the regulatory space and in the incentive space are going to be opportunities to get more samples, to better understand what's going on, and to look in places where we're not seeing cases yet.
Do you see in the future this being removed as a select agent? I mean, given that it's everywhere, endemic in wildlife on all continents.
Yeah, I mean, I think it is the question, right? So the culturing the virus is one thing, but who can do that, and how you can do that safely is one thing. But we dealt with that with mpox too, right? So changing the regulatory frameworks for who can manage mpox samples and how you can use them is something that allowed us to better find cases, and allowed the health systems to ready for more cases without worrying that they would completely functionally break down because they couldn't manage the waste or the samples safely. And so I think there is a potential that we could see short-term changes to who can work with H5N1 and who can't. I don't know that I would say that I would expect to see it completely removed from the select agent list. I think we have a lot more to learn about it. But once we start to see it everywhere, you can't, like, it's a meaningless, it's somewhat of a meaningless designation.
Well, there is an important issue here as well. Adolfo made that point about other species, because we're completely focusing on cattle at the moment. But there is, we have all the cases in the terrestrial, but also the aquatic mammals, all carnivores. And we see if the virus is being ingested, then directly the virus gets into the brain. The animals die before they can get the pneumonia. So that's a horrible thing. If that would be a mode of transmission in humans as well, then it would be devastating, of course. And that's why this whole issue of the milk is an important one. Yeah, but if you see how do the cats get infected, well, they probably got it through the milk, and so. I think that might be an issue. I think looking into other species as well, not becoming blindfolded towards the cattle. Yeah, because I think there's quite a number of other species. So, and then the birds is one thing, but there's a lot of mammalian species, mainly carnivores, as I said. But still, we didn't expect it in the cattle. But it is in carnivores, even you see transmission there. Yeah, if you look at the sea lions, etc., it's most likely it's being transmitted. So adaptation there, we have so far not found serious adaptation separate from the PB2 mutation. But I think there is an opportunity there for the virus to start further spreading amongst sea lions and the like. And humans do not get into contact with those a lot, but basically it can happen.
Right. I mean, but again, that's huge opportunity for additional transmission, because those carcasses are just rotting on the beaches and can be scavenged on anything, feral dogs, feral cats, birds. I mean, so where it goes from sea lions next is another question.
Yep. Well, especially the aquatic mammals is what worries me a lot, because they live in groups basically, and there's a lot of animal to animal transmission, most probably, although it has not been formally shown yet. But
we've been trying in South America. It's just really hard to formally show it.
I know. It can also be being infected from the same source.
So I think this is a phenomenal discussion, and in the interest of time, I'm going to move us to the last question, because I think it feeds into this point, in that how do we think that international collaboration and data sharing can be strengthened to facilitate a more coordinated global response, whether that's surveillance of these different modalities or global data sharing.
Yeah, I'll start with this because,
sorry. Go ahead.
I mean, I guess, we've been saying from the US perspective for a long time that we are expecting other countries to show transparency during outbreaks and share data and how important this is. And, I think it's really important that the US be a model of transparency during this outbreak if we're going during these negotiations on pandemic treaties to be expecting other countries to rapidly share data, and that this is such an important part of early pandemic response. And if we don't share sequence data, metadata, everything as transparently as we're expecting other countries to, there's going to be a lot of blowback down the line.
But we
Yeah,
go back
I completely agree with Marcia.
Right.
I think, just the fact that it's been a little less than a week and the pandemic treaty has almost failed primarily, well, maybe not primarily, but at least partially due to the pathogen sharing mechanism that's being discussed. So I think we have an opportunity in the US to step up and say, we're going to share in the way that we expect other people to share. And I'm not seeing that happen yet. But I can't agree more with Martha that there's an opportunity to make samples, make data accessible while we have time before we see this in humans.
But it goes also back to the earlier discussion, of course, the willingness of countries to share materials and data. I remember the whole story of the H5N1 in the early days, the problems we had with Indonesia and other countries. Yeah, just distributing the virus even through the WHO system. Yeah, and so basically we knew from certain countries long before it was declared that they were positive on the basis of some serology that had been done. Yeah, and so countries might not be very willing to share the fact that they have those kind of problems. Yeah. So it's the same.
But at least we should model what we hope to see in other countries.
Okay.
And the point is, if we can't meet our own expectations for other countries, how can we expect others to?
Who is we? Is that US or is
US? Yeah, if the US isn't meeting its own bar.
But we have seen it gives problems even there, yeah. So, and in Europe, it gives even bigger problems, you know. So even discussing now closing borders for poultry materials, you know. So this is crazy because we have a panzootic ongoing. Basically, it's in every European country. So closing borders is often an economic thing, yeah.
Yeah, no, it's a huge problem. I mean, and you saw even with France and, you know, when they started vaccinating their ducks and the US banned those products. I mean, none of this is science. So how do we get science to the forefront of these policy decisions?
Yeah. Well, that's a bad conclusion, I'm afraid.
Yeah. No, but the point is that
comes back all the time, yeah. I was referring before to BSE, but also the avian flu stories from the past. You know, it was always the same discussion. And if you look at Africa, yeah, during the COVID period, they were the first to report. They were the first to report new variants, and they had an enormous backlash there. Yeah.
Right, which was terrible for incentivizing reporting.
Yeah,
so I feel
absolutely.
like we're running into the same problems that we did during COVID in terms of punishing. You shouldn't be punished for reporting. Even in this cattle outbreak, we had individual states banning cattle movements from states that had reported positive H5N1. And so again, it was punitive. They were being punished for reporting. And we actually had farms we were working with who then saw that there were economic backlash against farms that reported and then said, we're not doing this anymore. And in some ways you can't blame them because if there's going to be an economic penalty for reporting data. So yeah, this was getting to what Lauren was saying. It all comes down to the whole policy, economic ecosystem and what these entities are incentivized to do.
But there's no proper international system to regulate it. The IHR is not being followed, you know. So I remember when we picked up MERS coronavirus, you know. So we should have reported that first to WHO, and so we left that to the Saudis, and that caused problems as well. So it makes it very difficult for a scientist, you know. So you want to know the epidemiology, but there's so many other factors that influence that.
I think this has been a fantastic and really insightful panel, and I'm really truly impressed by everyone's knowledge and expertise and dedication to the field. I think that our discussions, while at times harrowing, have illuminated a lot of the complexities and sparked some ideas for how we might drive solutions. And these type of insights are invaluable for enhancing global health security and pandemic preparedness, and highlighting the power of these types of collaborations and bold thinking as we discuss, and many different partners are listening. And so I think looking forward, we're going to reconvene in late May or likely June, and that'll allow us to dive deeper into diagnostics, vaccines, and other critical areas. But let's not forget, you know, we can push these boundaries, we can innovate and stay engaged and share these ideas. I think that we learn a little bit more each time, and putting science first to make new policies is a critical goal in pandemic preparedness. So I'm going to conclude with some resilience and persistent optimism. I hope you all stay resilient and stay persistent and innovating faster than an influenza virus can reassort. And I'm going to defer the last 30 seconds to our president, Sten Vermont.
Thanks, Maggie. I want to thank you for convening this panel on short notice. It was a bit of an experiment for the network to see how practical a quick consultation might be, and that the panelists went out of their way to make this hour free for us is amazing, and that nearly 60 participants were able to join again on short notice is also amazing. We hope we won't make it a habit of popping this on you, but it emerged from the discussions, the vibrant discussions of our respiratory virus action group from about 10 days ago. And there were just so many insights that some of you panelists had on that call, and some of the emerging questions that Maggie has shared with the group today, that we think this kind of rapid consultation may be something that GVN should be doing, and that we have a series. CDC wasn't able to participate in a big way on short notice, so that hopefully planning the next consultation panel discussion, CDC can weigh in, and we may be able to get some of the views of our European. We've sort of speculated on what should Europe be doing, what should the Africa CDC be doing in Latin America, what should we be doing, what about the Asian friends? We may be able to get some international perspective the next time around. So with that note, again, thanks to the panelists for your expertise, insight, and willingness to share, all of the participants who were able to join on short notice, and especially to you, Maggie, for pulling this all together and having the insight that this might be a helpful exercise. I've learned a tremendous amount in the last hour, and I hope even the experts have learned a little bit from the conversation and chit chat amongst each other, and some of the insightful questions that came from the audience. So again, thanks to all, and we'll look forward to seeing you in June.
Thanks all so much.